Today I will be talking about the genes and the DNA. And we have studied about the structure of DNA earlier in molecules of life in the classroom, if you remember. And even if you don't remember, then don't worry. We will see the structure of DNA once again. And I would like you people to know about the DNA very well, since DNA is one of the most important topic of biology. And you should know what that DNA is, what are the different components which are involved in the structure of DNA. Number 3, what are the functions of the DNA? So what is DNA? What is the structure of DNA? And last but not the least, what is the function of the DNA? So let me start with that understanding and using a very simple video. I just downloaded from the YouTube. It's very simple. Just if you click the genes in DNA, this might be the first video. And I'm just using the animation of the video to share my thoughts with you people. That every living thing, whether they are plants or they are animals, they are bacteria, or even the viruses, every living thing contains nucleic acids. And the nucleic acids are of 2 types. One type of nucleic acid is called as the DNA or deoxyribonucleic acid. The other type of nucleic acids is called as RNA or ribonucleic acid. But we are studying deoxyribonucleic acid as a primary topic today. So the DNA is part and parcel and the most essential component and molecule present in every form of life. Actually, DNA defines the nature of life. DNA defines the complexity of a certain life. DNA defines one life form will be different from the other life form. For example, humans are different from animals because the DNA, the deoxyribonucleic acids present in the animals, is different from the DNA present in humans. Animals are different from plants because they are having different DNA. So the amount of DNA and the nature of the DNA defines the nature of life form out there. So that is why DNA is considered as the genetic material. Earlier it was not known that actually DNA is responsible to transfer the genetic information from one person to another. So that is the DNA, which is actually controlling the almost everything in life. So if one organism or individual is different from the other, that simply means that that organism is carrying a different, either it's a different amount of DNA, or it's a different sequence of DNA, or it's a different type or nature of DNA. Even in humans, we are different from each other. Although we are carrying almost equal amount of DNA, almost, not 100%, almost equal amount of DNA, still you and me and every one of us in the class, we are carrying a different types of DNA. That is why you and me are different from each other. No 2 individuals carry the same sequence of DNA except in case of identical twins. In case of identical twins, the identical twins came from the one source, so that is why they have theoretically 100% same sequence of DNA. To make things a little bit simpler and easier for you, you should be able to see my screen as well as the YouTube video I have borrowed for you to explain you about the DNA. So it's there in every life form, plants, in animals, in bacteria, and even in viruses. It defines the nature of life. Humans also carries DNA in each and every of our cells. And we are different from other plants and from other animals because we are carrying a different sequence of DNA or different amount of DNA. And although the humans, they are like having the same amount of DNA, almost similar nature of DNA we are carrying, 99% of DNA between humans are like same, and only the difference of 1% make us different from each other. And that 1% difference lies in the sequence of the DNA. Only the identical twins, इसको monozygotic twins भी कहते हैं, the monozygotic twins, they have the same sequence. So that is something very special. There are something very special about the DNA. You can't understand life if you don't understand the DNA. The job of the DNA is that the DNA carries the genetical information, and that information is required to execute all the life forms. If you can recall my earlier lectures, how a DNA can control life, because life can only be controlled with the help of some functional molecules, and those functional molecules are proteins. DNA contains that information, that how different types of proteins can be created out of the sequence of the DNA through a two-step procedure. In the first step, a DNA is utilized to create a ribosomal RNA. A DNA is sequence of DNA is used to create a sequence of new RNA. That RNA is called as messenger RNA, or simply you can remember just as an RNA. And that process is called as transcription, the first stage. Creating RNA out of the sequence, out of the certain sequence present on the DNA. The next is using the sequence of RNA to create proteins. Can you identify this? This is like, these are the amino acids, these blue dots. These are the amino acids. So different amino acids merge together to create polypeptide chains or to create proteins using the sequence present on the ribosomal RNA. So this is the next step, using RNA and creating the proteins. This is called as, the second step is called as translation. This is called as translation. The first step here, synthesis of RNA, is called as transcription. And the second step is called as the translation. And together, this is called as central dogma. This is like everything about life. The most fundamental thing the life is carrying is a central dogma. Every life form has the DNA producing RNA, and RNA is producing proteins, and proteins are controlling the function. And that is also responsible to transfer genetic information to the next generation. For example, if the mother is having a brown hair kid, so the brown hair kid is carrying the genes which are responsible for the hair color from her mother. We got 50%, we received 50% genetic material from our fathers and 50% genetic material from the mother's side. So we are carrying the genetical information given to us from our parents, and that represents in us as well. That is why most of the genetical characteristics we are carrying, it came from our parents. We look similar to them in hair color, in eye color, because we are carrying almost similar sequences. But that's another problem. the genetical diseases are also transferred because of that problem. If the parents are carrying certain mutations, certain troubles in the sequence of the DNA, those troubles will also be transferred to the next generation as well. And they are called as genetical diseases. And every cell contains the DNA except that one cell. This is just an example of that one cell. If you remember the red blood cells present in the blood, they don't have the nucleus. That means they don't have the DNA as well. They have already a stock of proteins and RNA, which they can use to create all the functional proteins during their lifetimes. And that is why the life of the blood cell is the red blood cell is very small, just 90 days. The DNA by its structure is made up of a ladder. This is not the primary lecture to show you the structure of the DNA, but just recalling some of the things. It's like a ladder. The rungs of the ladder, which is सीढ़ियों के डंडे हैं, rungs of the ladder. They're made up of base pairs. This is base pairs. There are 4 base pairs which constitutes the DNA. And those 4 base pairs, if you can remember, those four base pairs are adenine base pair, guanine base pair, cytosine base pair, and thymine base pair. So these 4 base pairs present and create each strand of DNA. So this is one strand of DNA from this side. There is another strand of DNA present on the other side. So one strand of DNA with the base pair sequences, then there is another strand of DNA with the base pair of sequences. That is why it is called DNA is a double-stranded structure. Now each base pair is made up of 3 components. Each base pair is made up of 3 components. The one is a nitrogenous base, the second is the pentose sugar, and the third is a phosphate group. And together it is called as one nucleotide. So there are 4 nucleotides: adenine nucleotide, guanine nucleotide, thymine nucleotide, as well as the cytosine nucleotide. So one strand of DNA is made up of sequence of nucleotides. The other strand of DNA will create complementary sequence of DNA. What that complementary sequence of DNA means, that every A is only capable of making a triple hydrogen bond with a thymine. So every A meets the T, every G meets the C, every C meets the G, every C can bind with the G through double hydrogen bonding here and through triple hydrogen bonding here. Just some extra lines here, please, sorry. There exist 3 hydrogen bonds between G and C and 2 hydrogen bonds between A and T. These 2 strands runs anti-parallel to each other. You can see this is if the direction is here this way, the direction of this strand is this way. So if you know the sequence of DNA or sequence of nucleotide on one strand, you can automatically get the sequence of the other strand of DNA. Every A here will be complemented by T. Every C here will be complemented by the G on the other strand. And they bind and grow to create the double-stranded DNA. And that is how the structure of DNA constitutes all those base pairs. The sequence of these nucleotides defines the sequence of DNA. And sequence of DNA defines how we look. Because that sequence will ultimately produce proteins and to execute functions. And they exist in this double helical structure, which you can see. That is why DNA look like that. So hope this helps you to understand the DNA. Okay. And the next thing from the slides I want you to know, actually we have gone through a lot of things already about the structure of DNA. It has 4 nitrogenous bases. Each base is made up of 3 things. How these 4 bases actually connect. The DNA is originally reported to be discovered by these 2 gentlemen. I'm sorry about, I don't like these 2 people very much, but I should not speak like this as well. I mean, they are wonderful people. But the structure of DNA, the real hero, not hero actually, heroine behind the discovery of DNA was Franklin's. She generated the data using her X-ray experiments, X-ray crystallography experiments, and that data became a reason for these 2 people to create the 3-dimensional structure of DNA in 1950s, 1954, I guess, or something like that. These 2 will become very famous, and because of the discovery of DNA. Because before that time, we know that there is something which is controlling the function of life. That's like the God's stuff, some God's particle out there, something which is controlling the life. And we don't know the structure of that thing. And after that, it changes the life sciences altogether. After that, we started like taking science, especially life science, into very different directions. We came to know who is responsible chemically for the structure and function of life, that is DNA. The first thing we came to know about the DNA, oh, that is how the life is controlled. The second thing, we try to know more and more about the DNA, and the last thing is that we are trying to manipulate that DNA. We are now trying to change that DNA. Changing that DNA means we are trying to change the properties of an organism. We are trying to insert conditions. We are trying to insert new properties to the individual. That's what we are doing nowadays. So this is another question you should look at. I might be asking similar questions to you. So if there is every DNA molecule present in a cell should be transferred to the next cell. That is how we can forward the information from one cell to another. So if this is like supposed to be the original, the old, or I must say the original DNA, if this is the original DNA, this one, before the cell division, this should be duplicated. So one of the strand came from the original DNA, the other one is newly synthesized. So this one is the old strand and this one is synthesized during the process of cell division. This is called DNA replication. So we create copies of DNA so it can be entered into the new cell. So every cell receives the same sequence of DNA, same amount of DNA as it was present in the parent cell. Okay, so DNA, how DNA works, a portion of the DNA, a portion present on the DNA is called as the gene. It is called as gene. This is a term which you heard in different sci-fi movies and in other literature as well, gene. The person has a wonderful genes, the person has a different type of genes. That is a gene belongs to a particular disease. This is the gene belongs to the height of the person. This is these are the genes which are responsible for eye color. One gene is portion of the DNA which carries an information to produce a protein. So one part or one portion of the DNA which can produce a protein is called as the gene. So like one gene is able to produce an RNA, as I showed you earlier, and that will produce a protein. So once the RNA is created within the nucleus, this blue thing is the nucleus. Once the RNA is created within the nucleus, it is exported from these nucleopodes into the cytoplasm. This copy of messenger RNA is created out of the DNA from the portion which is called as the gene. And then using this sequence, the next step is the protein synthesis. Within the ribosomes, as you know, it is responsible for protein synthesis. The sequence of messenger RNA is used as a template, and using that information present on the sequence of messenger RNA, which is transcribed from the DNA earlier, using that information here, a ribosome creates and puts amino acids together. First amino acid, second amino acid, third amino acid, fourth amino acid, and that is how it creates a polypeptide chain or a protein. If you know the sequence of DNA, you can create the sequence of RNA as well. There is some little difference between the DNA and the RNA you should know. DNA is double-stranded, RNA is single-stranded. First, very important. DNA is double-stranded, RNA is single-stranded. The RNA does not contain the thymine. So instead of thymine, the RNA contains another base pair which is called as uracil or U. That's one difference you need to know. DNA is deoxyribonucleic acid. The RNA is ribonucleic acid. The pentose sugar present in the DNA contains one oxygen less. That is why it is called deoxyribonucleic acid. The pentose sugar present in the RNA, it contains normal atoms of oxygen. It is called as ribonucleic acid. So if you know the sequence of DNA, for example, this is the sequence of DNA, you can generate the sequence of RNA as well. The sequence of RNA is complementary, but the sequence of RNA does not contain the T. So for example, if there is an A, there should be, let me write the sequence of RNA here. If there is an A here, it should be complemented by every A complemented with the T. But since the T is not present on RNA, so I am writing U here. Every T is supposed to be complemented with the A. So every T will represent on the RNA with A. Every C will be represented by G, and every G will be represented by a C. Every T will be represented by an A, and every A will be represented by the T. But since the T is not present, so I am writing here U, which is a different base pair present in the RNA. So these are the very simple questions which some people might be asking to you. So if a sequence of DNA is changed, if there is something bad happens with the sequence of DNA or the sequence of the genes, you can guess the gene sequence will generate the RNA. The RNA will going to be used to create proteins. If a change is there in the sequence, in the normal sequence of the gene, this type of change is called as mutation. This is called as mutation, the change. The change may be substitution, it may be insertion, or it may be deletion. So if a change is there in one base pair, if a sequence is changed, you can guess the RNA will be different. If the RNA is different, that automatically means it will create a different protein or maybe no protein at all. So any damage in the sequence of the DNA or any damage in the sequence of the gene will result in a misinformation on the messenger RNA, means it will lead to a faulty protein at the end of the day. Or maybe the cell will lose the function altogether. So mutations in the genetic material will result in the trouble in the structure of the protein. It may be an additional base pair, that is a mutation. If a base pair is added, if a base pair is removed, that might be a mutation. So all these mutations changes the DNA sequence, that might be good or that might be bad. Maybe the change results in the creation of a better protein with the improved function. That is how humans or evolved over the period of all those millions of years by slowly changing the mutation with the sequences of DNA present in life. It results in small change, creating variety of organisms. Some of them have improved functions, some of them are malfunctioned. The improved functions, the organism with the improved function, they have a better chance to survive, and they dominated the life forms and they survive over the period of all those time. It's a slow process, ongoing process, and that resulted into a better and better organisms over the period of time. So whatever the structure we are carrying is actually a result of all those mutations which occur during all those millions of years. One single mutation can create havoc, can cause a lot of trouble. For example, this is a disease called sickle cell anemia. It's a very deadly disease. The person or the baby is not able to, the baby's blood is not able to absorb oxygen well because of the error in the gene which create hemoglobin. Hemoglobin is a part of the blood cell. Hemoglobin is a protein, so it is created from the information present on the DNA. Unfortunately, the GAA sequence, which produce an amino acid called glutamic acid, so this 3 base pairs is called as codon because it produces an amino acid. At the end of the day, there was an error that A replaced with the T. This small change in one base pair created a different codon, and instead of glutamic acid, this sequence incorporated valine. So whatever the new hemoglobin is now producing, we have a new hemoglobin here, and that new hemoglobin will not be able to absorb oxygen efficiently, and that is the trouble so mutations can cause this trouble. A lot of other diseases are also a result of the mutation, particularly cancer. Over the period of our lives, we develop a lot of mutations, and some of those mutations disrupt the function of certain important proteins in our body. Some of the mutations disrupt certain important functions of the cells. Cells became mad. They started dividing rapidly and eventually producing lots of cells which are not required, and that's what we call as cancer. Okay, so mutations are supposed to be quite harmful. So this is our lectures today. Thank you very much for listening.
सर आप लास्ट टाइम बताया था कि अगर डीएनए को मैनिपुलेट करना हो या ट्रीट करना हो तो वो इतना ही मुश्किल है जैसे आप स्पेस स्टेशन से 5 रुपए का कॉइन एफसी के ग्राउंड में स्ट्राइक करें.
बिल्कुल सही
सर, इस बारे में थोड़ा बताइएगा. ये काफी चीज फैसिनेट करती है, कैसे करते हैं, तो ये बताइएगा कि क्या करते हैं हम इसको करने के लिए.
ऐसे मौके पे आमतौर पे पता है अब मुझे क्या कहने को दिल करता है, वैसे वो बात है थोड़ा शौक होने वाली. यू शुड रेफर टू माय वीडियोस प्रेजेंट इन माय चैनल. ओके लेकिन अफसोस की बात ये है, टॉपिक का नाम है टारगेटेड जीनोम एडिटिंग. वो 4 लेक्चर्स हैं लेकिन वो 400 लेवल के हैं. बट यू कैन सी के हमने कैसे इवॉल्व किया. ये एकदम से नहीं हुआ, हमने तरतीब से सीखा और आज हम वो कर पाते हैं जो पहले हमारे लगता था कि ऑलमोस्ट इम्पॉसिबल है. तो ये स्टेजेस और स्टेप्स के साथ हुआ है, ठीक है, लेकिन ये डूएबल है.
सर मैं ये पूछ रहा हूँ कि आया कोई लाइफ मिली है जिसमें डिफरेंट बेस मॉलिक्यूल्स हो डीएनए के अंदर, अगर होंगे तो उससे क्या असरात पड़ेंगे?
मेरा आपका सवाल शायद इंटरनेट की वजह से मुझे अच्छे से मिला नहीं है. आप एक बार और फ्रेज करें, मैं सुनने की कोशिश करता हूँ.
सर मैं ये पूछना चाह रहा हूँ कि डीएनए के अंदर जो बेस मॉलिक्यूल्स हैं, सर वो आया सारी लाइफ फॉर्म्स जो मिली हैं हमें अब तक उनमें सब में सेम है या आया कुछ ऐसी लाइफ फॉर्म्स भी है जिसमें बेस मॉलिक्यूल्स डिफरेंट है?
ये बहुत अच्छा सवाल है.
डिफरेंट होने से किस तरह के असरात आ सकते हैं?
ये बहुत अच्छा सवाल है और ये बहुत बड़ा सवाल है. अब तक हम जितनी भी लाइफ फॉर्म्स को जानते हैं उन सब में यही वाले 4 बेस पेयर्स होते हैं. दे कंटेन द सेम 4 बेस पेयर. नाउ द नेक्स्ट क्वेश्चन इज दैट क्या 4 बेस पेयर की बजाय 5 बेस पेयर वाला डीएनए हो सकता है? और इनोवेशन करें दिमाग में. Is that possible? Instead of 2 strands of DNA there might be 3 strands of DNA. if we can think in this way, that is there any other way of creating genetic material? I believe there exist other ways of creating genetic material. We don't have any. There is no genetical material other than the thing which we learned with the 4 base pair. What I believe is that when some aliens came to our planet in the future, you know what they will be looking at? They will not be looking at the mines. They will not be looking at the sona and gold and platinum and uranium, because these things are present everywhere in the universe. Every other star is carrying all this. What we never found in any other planets, on any other planets, on any other distant galaxies and on the other stars, is the DNA and the proteins. These are very unique molecules to us. We never observed any molecule like this anywhere else on the universe. So I believe the aliens will be interested in our DNA, and if they come, they will be interested in our DNA. There exist many sci-fi movies who use that idea that if, like, instead of the 2 strands of DNA, if there are 3 strands of DNA, the level of complexity will be huge, and we might have a very different life form. If, like, base pairs, instead of 4, there might be 6 base pairs, there might be 8 base pairs. What will happen? The complexity in the life forms will be different. Who knows? The other life forms, they might be having entirely different, type of genetic material. But in Earth and in all of our human history, we never found the genetic material like this anywhere in the outer space. Hope this answered the question. G.